Hello friends, welcome back to session second. We were discussing the questions of GATE Biotechnology 2020. Up to now, we have discussed the first 25 questions related with the biotechnology. Each question was of 1 mark. In this particular session, we are going to discuss a question left behind questions, approximately around 20 questions they are there, and here each question carries 2 marks. So do subscribe our channel IFAS Online Life Sciences. So always you remain alerted. We will upload various other videos related with the different examinations. So be tuned. You can also visit our website ifasonline.com. So now readily moving to the question. This question is basically the changes in the temperature of water when a water is in a iced form to a steam form. Which of the following profile is correct, or you have to identify which of the following trajectory is correct. So let's go through this question, try to analyze this. A block of ice which is at 0 degrees centigrade. It is supplied heat at a constant rate to convert ice to a superheated steam. So what we want to do is to convert it into a superheated steam. Which of the following trajectory correctly represents the trend of temperature of system with the time? Here you have to take that the specific heat of water is not a function of temperature, means it is a constant throughout the temperature. Now what happens is that since water is at 0 degrees centigrade, so let's say this temperature is 0 degrees centigrade, although water is at 0 degrees centigrade, but it is not in a liquid state, it is in a ice form. So as you increase or you will provide a heat, there is a need of an energy which is required for a conversion of a water molecule from an ice form to a liquid form, and this energy which has to be provided is referred to as molar heat of fusion. That is, first there is a need of an energy where the temperature do not change, but this energy is required for basically disrupting the hydrogen bonds present in ice water to convert into a liquid state. This energy or heat which is required from outside for converting this ice into a liquid state is referred to as a heat of fusion. We can also define a heat of fusion. Heat of fusion is the amount of energy which is required for conversion of 1 mole of ice into 1 mole of water at its we can say melting temperature or freezing temperature. But once ice has been converted to a liquid water, now further providing an increasing a temperature, what you will observe is that there would be a constant trend in the increase in the temperature of water. So with time, as you are heating with the time, there would be constant increase in the temperature that depends upon a specific heat, and it is being said that the specific heat of water is not function of temperature, so there would be a constant increase, and this constant increase it would be seen up to 100 degree centigrade. So specific heat, as you know, that it is amount of heat which is needed for a 1 gram of water to 1 degree centigrade is a constant, that is around 1 kilocalorie energy would be needed for a 1000 gram of water. Up to now there is no change. Now water, liquid water, here it was ice which was melting into a liquid. Now the water, still it is in a liquid form, it has not been converted to a vapor form. Now again you need an energy over here, so that now water which is at 100 degrees centigrade could be converted into the vapor form. This energy which is needed from outside is referred to as molar heat of vaporization, where the temperature will remain same, that is around 100 degrees centigrade, but temperature will remain same. Further, if you move beyond this temperature above 100 degrees centigrade, and now you can see that end of this line, the water has from liquid, it has been converted into steam. Then as you increase the temperature, as you further heat, there will be a straight line increase in the temperature. So graph would be somewhat like this. So this cannot be a graph because initially here heat of fusion has not been seen. It is not, this is not the case. This would be the correct case. Here the heat which is required is referred to heat of fusion, and here are around 100 degrees centigrade. Here you need an heat which is referred to as heat of vaporization. So this is a question number 26. Now moving to question number 27. This question is related to the PCR. The template DNA is being given, and you have to identify that which would be the appropriate primer for the amplification of this particular kind of DNA. So you can easily make it out. Let's say you can easily identify that what would be the forward primer. So forward primer, which will align over here, its sequence you can easily identify. 5 prime, it would be G, C, T, A, G, A, somewhat like this. So G, C, T, A, somewhat like this would be the primer on this side. And the primer which is going to be on this side, which would be again 5 prime to 3 prime, it will have a sequence like G, C, G, C, T, and so on. So second primer should be G, C, G, C, T, A, so on. Here this is not the case. So correct answer for this choice would be answer B. Moving to next question. Question number 28. Some statements they are given about the immune response, and you have to identify that which sub-statement is correct. First statement is T cells are activated by antigen presenting cell. Yes, that's true. T cells, they are activated by an antigen presenting cell. That's true. From antigen presenting cell, that it could be a macrophage, dendritic cell, or even B lymphocytes. That's true. That is, these antigen presenting cells, they will display a processed antigen to this, and this processed antigen would be recognized by the T cells. That is true. And this is an antigen presenting cell. Foreign peptides are not presented on helper T cells by class 2 MHC. No, this is false, because you know that foreign peptides, if they are being taken by a macrophage, they will get processed into the lysosome, and once they have been processed into lysosome, they would be packaged into vesicle, and they would be expressed on the surface in a bound form with the MHC class 2. So this is a vesicle which is bringing the MHC. So this statement is false. And if this statement false, you will have to be end with one choice only. Our statement says that dendritic cells are referred to as professional antigen presenting cell. Yes, that's true. The cells which expresses an MHC class 2 are often referred to as professional antigen presenting cells, and professional antigen presenting cells include mainly B lymphocytes. It includes macrophage, and one more professional dendritic is the dendritic cells. These are the professional antigen presenting cells. Moving to question number 29. Which of the following statement is correct about the eukaryotic cell cycle? Let's go through these questions one by one, statement one by one, and try to find which statement is true. First statement is Cdk can phosphorylate protein in the absence of cyclin. No, Cdk, they need a cyclin for their activity, so this statement cannot be true. And if you know this, then you have to end with only one choice. But let's go through the other choices as well. Cdk can be inactivated by phosphorylation. Yes, if there is an inhibitory phosphate, Cdk would be inhibited, and if there is an activatory phosphate, even it could be activated. So this statement is true. Degradation of cyclin is required for cell cycle progression. Yes, we have studied in a class that cyclin B, if it get degraded, then only cell will progress from an M phase back, or it will able to exit an M phase and will able to enter into G1 phase. Cdk are not involved in chromosome condensation. No, they are involved in a chromosomal condensation, because we know that MPF, which is a complex of CDK1 cyclin B, it is involved in the compactation or condensation of chromosome. So this is false. So correct statement are Q and R only. Moving to question number 30. In this case, W, X and Y, these are the intermediate of conversion of some substrate into the products. So the pathway is already given, W, X, Y. This is the same in all the options. You have to identify that there are 4 mutants, Z1, Z2, Z3 and Z4, and at which step actually this mutant is working. So you can easily make it out. How? Let's see. Analyze this. Z1 mutant, if you provide W or if you provide X, they do not grow. But if you provide Y or Z, they can grow. Means if you provide Y or you provide Z, they will grow. Means this path must be mutant in case of Z1. So this step should be mutant in a Z1. So if you provide W or you provide X, it will not be converted to a Y. So Z1 should be here, and if you know this only, then also you reach to this conclusion. Similarly, Z2, you can see that if you provide W, it doesn't survive or grow on medium, but in X, Y and Z it can grow. So it means it should be at this particular place, that is between W and X. Here there must be a Z2, this would be the answer. Similarly, you can see it is not surviving in case of W, X or Y, but survives in case of a Z. Means just prior to Z, this should be mutant. Z3 should be mutant. While Z4, it is able to grow in all conditions. This enzyme must be mutant, where the Z next of steps they are functioning properly. Then W could be given to X, X could be given to Y, or Y could be given to Z. So correct answer would be option 2. Moving to next question. This is basically a cross, and results, they are being shown for an F1, and in tomato plants, red is dominant over, okay, red is dominant over, first it is given that red R is dominant over, we can say yellow, and for yellow the genotype is small r, while fruit color, purple, that is capital P fruit color, it is dominant over the small p. For a green, purple is a dominant over green, which is represented by small p, green. Now, the number of progeny of plants, different fruits were meeting as follows. Means that this is a result of some cross. It is a result of some cross. And what you can see that in this case, if you break it into this dihybrid cross into monohybrid cross, what you find is that red fruits are 145 plus 184. While the yellow fruits are 66 plus 47, that is 5 plus 4, that would be 9, 8 plus 4 would be 12, and you will get a 329 over here, 1 over here, and here you are going to 1. So approximately for fruit color you are getting a ratio of approximately 3:1, and if ratio is 3:1, it means the genotype at this locus for the both parents should be capital R small r into capital R small r, because only under this case you are going to get a ratio of 3:1, that is red into yellow. In case purple stem is 145 plus one more purple stem is 66, and green stem is you can say 184, and another green stem is 47. Adding these together, what you are going to get is 6 plus 5 is 6 plus 5 is 11, 6, 7, 4, 11 over here, 211 over here, 7, 4, 11, 4, 5, 3, something like that. So approximately what you are getting here is a ratio of 1:1, and if you are getting a ratio of 1:1, this simply suggests that the genotype of this particular locus should be capital P small p into small p small p. It should be like a test cross, which could give only can provide you ratio of 1:1. So genotype of the, on basis of this information, we can say that genotype of this locus should be heterozygous at both the cases, heterozygous for both the cases, heterozygous at both the locus. And for second locus, there must one of plant should be heterozygous, another plant should be homozygous, one should be heterozygous, another should be homozygous, one should be heterozygous, this is also heterozygous, so this cannot be answered, this cannot be answered, this cannot be. So correct answer would be option 1. Question number 32, this is a direct straightforward question. In this case, you have to identify the cytokinins which are synthetic analogs. Means there are some auxins which are naturally present, and there are some auxins which are synthetically produced. Which of the following are synthetic? So BAP, yes, BAP is one of the first synthetic auxin which is being commercially utilized, benzyl aminopurine. So this is synthetic, P is a synthetic. Then zeatin is one of the naturally occurring cytokinin in coconut milk and various other plants. Kinetin, as you know, Skoog produced this kinetin through the hydrolysis, or we can say autoclaving of sperm DNA of a whale. So this is also an synthetic. So synthetic would be P and R. 2-isopentyl adenine, it is also natural occurring auxin present in various woody plants. So answer would be BAP and kinetin, both are synthetic. Question number 33, this is also direct straightforward. Question is that Carl Woese used gene sequence of which of the following for a phylogenetic relationship? So this direct question, you know that the Carl Woese, it has utilized 16 srRNA for phylogenetic relationship. On basis of this 16 srRNA, he has divided all the organisms into 3 groups. That is, one is Eobacteria, and Later on, he divided further into two more groups. One is archaebacteria and eukaryota. But this classification was based on 16S rRNA, and 16S rRNA, it is present in the small subunit of ribosome. So answer would be a ribosomal RNA, that is 16S rRNA, and the 16S rRNA is present in a small ribosomal subunit. Moving to question number 34, this is a direct straightforward question. Here some pathogens are disease causal organisms, they are given, and here some drugs which can be utilized against them. The drug which is often utilized for the treatment of influenza A virus is the amantadine. A must be 2. For fungus, you use a fluconazole. This is also true. For plasmodium, which causes a malaria, you can use an artemisinin. For mycobacterium, which leads to tuberculosis, the drug of choice is isoniazid. So this is a direct straightforward questions. We have already discussed the even mechanism of all these 4 drugs during our online classes. Question number 35. There are some assertions and reasons. You have to find out whether assertion and reason both are true or not. DAM methylase protect from E. coli from a phase endonuclease. No, DAM methylase, you know that it is responsible for maintenance methylation. It is responsible for maintenance methylation. That is, there are some specific sites, GATC, they get methylated once the DNA has been replicated, short while after DNA replication. So it is not playing a role in a restriction modification system, it is playing a role in a maintenance methylation. So this is false, A is false. E. coli DAM methylase methylate the adenosine residue in the sequence GATC. Yes, that's true. That is adenine, it is being methylated at adenine residue. So A is false, R must be true. A is false, R must be true. The correct answer is option G. Question number 36. Again, it is related with an assertion and reasoning. Some assertion is given and you have to identify whether the reason is true or not. Let's go through this. Embryonic stem cells are suitable for developing knockout mice. Yes, embryonic stem cells, they are rapidly dividing cells. Here a homologous recombination would be functioning. So that's why they are preferred for developing knock-in, knockout or even transgenics. Homologous recombination is more frequent in embryonic stem cells than in somatic cells. Yes, that's true, because the somatic cells do not divide, and the chances that they will be in a stage after S phase would be very lesser, because most of the somatic cells, they remain arrested in G1 stage, and under this scenario the chance of homologous recombination would be very rare. So we can say that A is also true and R is the correct reason for this. Answer would be option B. So let's, friends, move to the next question. In next question, here you can see that what the schematic plasmid with a gap in one of the strands is shown. So here there is a gap. It is being shown. Which of the following you have to use for filling this gap? So since at 5' there is a OH group, first you have to add a phosphate group over here. You need a phosphate, that would be by a enzyme polynucleotide kinase. So you need a polynucleotide kinase. 3' OH group, it has to be extended. So for this you need a DNA polymerase. And finally, this extended 3' should ligate with a 5' phosphate group. So for this, we need a DNA ligase. So what are needed for filling this gap? So you can say that here DNA ligase is required. Yes, it is required. Alkaline phosphatase? No, it is not required, because already at 5' end there is a OH group. There is no need to remove a phosphate group. This is not required. DNA polymerase is required and polynucleotide kinase is required. Yes, so answer would be P, R and S. Moving to question number 38, you have to simply match this and you have to reach to a correct conclusions. So mitochondria, P could be either 2, that is double membranous organelle, or it could be explained by an endosymbiotic theory. So you have left mitochondria, or even it could be explained by this 2, 3 or 4. Means mitochondria can show maternal inheritance, it could have endosymbiotic origin, or even it could be a double membranous organelle. So 2, 3, 4 could be for P. So this option has been removed and 4 is not given, so 4 is not there in option. So for mitochondria it would be either 2 or it would be 3. 4 is not possible because it is not given in the options. For chloroplast, again, there it could be double stranded structure, it could be a maternal inheritance, or even it could be endosymbiotic origin. Again, it could be 2, 3 or 4. 2, 3 or 4. One cannot be there, so even this has been rejected. So this could be 3, this could be 4. Okay, that's fine. This could be 3, this could be 4, or even this could be 2 or this could be 3. This could be fine. Nucleus, for nucleus there only one thing is possible, and that is a double membrane enclosed structure. The nucleus is a double membrane structure. So it would be R must be 2. R cannot be 4, that nucleus is endosymbiotic origin, that could not be true. And so 3 is okay, 4 is okay, 2 is also, and for endo ER, only one thing is possible, and that is single membrane structure, so it would be 1. So correct answer for this, out of the given choices, would be option C. Question number 39. Some regulatory strategies are given for the enzymes, and you have to identify that which of the regulatory strategies is correct. Phosphorylation yes, there are various enzymes which are regulated by phosphorylation, dephosphorylation. For example, glycogen synthase, it become inactivated by phosphorylation, while the glycogen phosphorylase, it get activated by phosphorylation. So this is true. Allostery, yes, this is one of the most common way of regulation of enzymatic activity. We have studied in the class the case of ATCase, aspartate transcarbamylase, being activated by ATP or GTP, inhibited by CTP, or you can say UMP or CMP. Feedback inhibition, yes, that is also one of the way of regulation which is seen in allosteric enzymes. So these all three regulatory strategies, they could be utilized for metabolic regulation. So correct answer is P, Q and R. Moving to question number 4, T, where again some assertion is given and some reason is given. You have to find out whether assertion and reason both are true and there is a correct explanation for this or not. Let's see. A zygote and its intermediate distant cells, means let's say 2 cell stage, these are intermediate distant cells, are unspecialized, yes, that's true, and are called as totipotent. Yes, this A is true. Totipotent cells remain the capacity to differentiate into only few. No, totipotent cells, they do not differentiate only few, they have a total potential. Total potential means they can convert into a complete organism. Yes, they can convert into complete organism. So they are not having only potential to differentiate only few cells. So R is false. So correct answer should be that assertion is true, but its reason is false. Then moving to question number 41. Again, there are some techniques which could be utilized as a gene therapy for in a germ lines or in a somatic cells, and you have to identify that which combination is correct. So statements are related with the gene therapy. Affected individuals but not their progenies can be cured through the germline therapy. No, germline therapy means transferring the genes into the germ cells which are going to produce gametes. This means it could be inherited even to next generation. So this statement is false. P is false. Affected individuals as well as their progeny can be cured through germline. Yes, that's what we are saying. The affected individuals as well as the progeny, they could be cured using germline gene therapy. That statement is true. Affected individuals but not their progeny can be cured through a somatic gene therapy. Yes, that is also true, because the information which is present in somatic cell is not going to get passed to the next generation. Only the organism where the gene therapy has been made in a somatic cell, that would be get benefited by this particular approach. So R is also true. Affected individuals as well as their progeny can be cured. No, progeny cannot be cured. So correct answer would be Q and R. Moving to question number 42. Again, the question is related with the assertions and reasons. First assertion is a genetically engineered rice, means we are talking about a golden rice, that produces a beta carotene is in rice grain is called as golden rice. Yes, we have studied a complete story of Ingo Porticus, that how he made it possible, that how he made the genes from daffodil and later on from maize as well as from Irvinia crotovaria, and he was able to produce beta carotene even in the rice, and that rice has been named as golden rice. Enabling biosynthesis of provitamin A, which is beta carotene, is also referred to as provitamin A, in the rice gives a characteristic yellow orange color. Yes, that is true. That's why this has been named as golden rice. So correct answer is that both A and R is true, and R is a correct reason for A. So correct answer would be option C. Moving to question number 43. Basically, this is a probability based question. It has been said that there is a sequence of 1 megabase pair long. The DNA has all 4 bases occurring in equal proportion. That is probability of any base, chance is 1/4. That is all, the probability is same. The number of nucleotides on average between the 2 successive equar recombinant sites, that we have already discussed in classes, that the chance of G at the first place is 1/4 because probability is same. Of this is also 1/4. The probability of another T is also 1/4. Another T is also 1/4. So the chances is getting the sequence is 1 in 4,096 base pair. So if one sequence is over here, one site is over here, then another site is expected to be spaced by 4096 base pair as per the probability, assuming that all the sequences, they are equally distributed. Moving to the question number 44. in this question, it is being said that initially the E. coli was being maintained on an N15 containing medium for several generations. So we can say that, assume that both the strands here over are in the DNA is heavy. Cells were then transferred to an N14 medium and were allowed to go for 4 generations. The proportion of total DNA with an intermediate density that has to be calculated. So let's say the same DNA strand, it has been transferred to first generation, then it is being transferred to second generation. This radio will remain same. The proportion of the DNA with we can say heavy isotope will remain same. All the new synthesized DNA would be the DNA having a nitrogen 14. So after first round on placing with an N14, what will happen is that you will get 2 hybrid, that is this would be also of intermediate density, heavy light and heavy light. In next round what will happen that this will also open and this will also open. Newly synthesized DNA would be of light strand type. So again now you are getting a 2 heavy light and rest of 2 would be light. In next generation what you will find is that all these will open. Let's say these all get open. In this could be explained like this. Again, now what you will observe is this would be newly synthesized DNA. Now you will have 2 heavy light and rest of all would be, you can say 6 of them would be light. So here you are getting a 2 DNA strands, both of them are of heavy light. Here 4, here you are getting 6, and here you are going to get 4, here you are going to get 8, and here you are going to 16. Out of 16, these only 2 will be heavy light and rest of the 14 they would be light, and you have to calculate a proportion, and proportion would be total strands you are going to get 16. From 16 to proportion would be, it has to be calculated the proportion of the total DNA. So proportion of total DNA could be calculated is 1 by 8, and 1 by 8 proportion would be 1 by 8 proportion. You can say that in percentage it would become out proportion would be 0.125. in percentage if you convert it will get a 12.5 percentage. So if you representing it in percentage it would be 12.5, but proportion would be 1/8, and 1/8 proportion means it would be 0.125. Then in question number 40, a batch reactor is inoculated with a 1 gram liter biomass. Under these conditions, cells exhibit a lag phase for 30 minutes is lag phase. Then there is a specific growth rate in log phase which is 0.00417 per minute. The time taken of biomass to increase from 8 gram per liter to 8 gram per liter would be, you have to calculate this. So we know that doubling time, that is G, is equivalent to log 2 by maximum specific rate, maximum growth rate. Now you put it over here, log 2 divided by maximum specific rate is also given. The value of log 2 is 0.693. The maximum specific rate is also given. So you can say doubling time is 166.22 minutes. And what you have to do is that now 1 gram it has to be doubled into 2 gram, 2 gram has to be doubled into 4 gram, and 4 gram it has to be doubled into 8 gram. So you need at least 3 generations. So total duration which would be required in a log phase would be 166.22 into 3, that will come out to 498.66 minutes of log phase. But as it was said that prior to log phase there is also a lag phase of 30 minutes. So 498.66 minutes of log phase plus 30 minutes of lag phase, if they are added to it, you will get 528.66 minutes. So correct answer would be 528.66 minutes. Moving to question number 46, this is a question related with the maths. That is, the system of linear equation is given, cx plus y is equal to 5, 3x plus 3y is equal to given. It will have no solution when the value of c, if you place a value of c equal to 1, the equation will become x plus y equal to 5, and you will get 3x plus 3y equal to 6. Now you cannot solve this. So if you will place a value of 1, it will not have any kind of solution. Moving to question number 47. It is being given that codon for phenylalanine is 2, for codon for leucine is 6, codon for isoleucine is 3, codon for methionine is 1, codon for serine is 4. What would be the total number of unique DNA sequence that encode this peptide? So this is the peptide. So simply you have to make a multiplication of this. For phenylalanine there are 2 codons, for leucine there is 6 codons, so it would be multiple of this. For isoleucine there are 3 codons, for methionine there are 1 codon, for serine there are 4 codons, and again for leucine there is a 6 codons. If you multiply it, what you will get is 864. So 864 possible DNA sequences that can encode this kind of polypeptide. So combinations of the codons will give you the combination of the DNA sequences which can code this kind of polypeptide chain. Moving to question number 48. Now it is being said that assume that cell cultures was started with a 5 human fibroblast cells. So there were 5 cells from where we are starting. 2 cells did not divide. Means 3 cells they are dividing. 2 cells they have been left behind that they do not divide. The 3 cells, they completed 3 divisions, 3 rounds of division. Means after first round of division, there are 6 cells. After second round of division, there would have been 12 cells. After third round of division, there would be 24 cells. Now total number of cells, which would be 24, which is after the division, and 2 which are non-dividing cells. Total there are 26 cells. Now at this stage, the total number of kinetochore in all cells put together would be, so we are assuming that these cells, all of them, they are in a G1 stage, and at G1 stage there are 2n is equal to 46 chromosomes in our humans, and each chromosome, if it is in G1 stage, that is having a 1 kinetochore. If it would have been a post S phase, or we can say in M phase, then each chromosome will have a 2 kinetochore. But the cells, just after completing the division, they would be in a G1 stage. So we are assuming that one human cell has a 46 kinetochore, that is 1 kinetochore per chromosome. So there are 26 cells. Each cell has a 46 kinetochore. If you multiply it, you will get 1196. So these must be the possible number of kinetochore, assuming that all cells after division, they are in a G1 stage. Question number 49. This is the question where you have to calculate the concentration of biomass in a chemostat at a steady state. For this, the specific growth rate is given, the Ks is also given, that formation of substrate as compared to product utilization rate is also given. You have to utilize this particular we can say equation to assess the biomass at a steady state. If you put these all values, that is, this is the actual specific rate, this is maximum specific rate, this is specific rate observed, this is Ks value, and if you put all this substrate input, which is also given that you are adding this 20 gram of glucose at the rate of 1 liter per hour, you will get the answer around 0 gram per liter. Going to question number 50. A function is f is given as fx is equal to 4x minus x square. The f value is maximized if x is equal to obviously 2. You can simply go through this. Let's say you put a value of 1. So function of 1 would be 4 into 1 minus 1 square, that will come out to be 4 minus 1 square is 1, that will come out to be 3. If you put a function of 2, what you will get is 4 into 2 equal to 2 square. What you will get is 8 minus 4, the value comes out to be 4. So it is 4 is greater than this. Similarly, if you move to a function of 3, again the value will start declining. Function of 3 would be, you can say 4 into 3 minus 3 square, that will be 12 minus 3 square would be 9, that will come out to 3. So 4 is the maximum, again value is declining. Similarly, if you go for function of 4, you will get 4 into 4 minus 4 square, that would be 16 minus 16, value will come out to be 0. So maximum value will be for a 2, because it will show this curve like this. Again the value will start declining. Maximum value we will see at the 2. Question number 51. This is a case of infinite series where the values of S, they are given 1, 2, 3, 9, 4, 27. So here 1, 2, 3, 4, 5, 6 and so on. And here there is a multiple of 3, 9, 27, 81, 243 and so on. The value, you can add it. The value of 1 of this, 2/3 is 0.66, 3/9 is 0.33, 427 is this. If you will keep on adding that after certain length, that is after the 2 digits, the changes is negligible. So if you even add over up to here, what you will get, the value would be approximately 2.22, and further the values will keep on increasing, but that will make a difference only in this you can say third or fourth digit. It is not going to make any difference, and so correct answer would be values will range from 2.2 to 2.3. Moving to the next question. This question is also straightforward. There is a protein A and protein B. Let's say this is a protein A and there is some protein B. Form a covalent complex. The gel filtration chromatography of this compound shows a peak of course, means this complex from gel permeation chromatography, it is being found as of 200 kilodalton. Means it could be monomeric proteins, these could be a dimeric protein, we do not have any idea. Even they could be of tetrameric protein, we do not have any kind of idea. Every protein could be of 2 subunits, 4 subunits and so on. But the total mass of this is also 2 kilodalton. Complex of mass of this is 200 kilodalton. Then SDS page analysis of this complex, in absence of beta mercaptanol was found to be 50 and 25, while in presence of beta mercaptanol it was at 25. So only single band, and it is given that molecular weight of A is 25, then molecular weight of B of each subunit of B should be also of 25 kilodalton, because if it would have been a different molecular weight, then in presence of beta methanol you will get a separate band. You are getting only one band, that also of 25 kilodalton, which simply suggests that protein B must be also a 25 kilodalton. So real scenario would have been like that, protein A is also composed of 4 subunits, 25, 25, 25, 25, while the B protein is also composed of 4 subunits of 25 each, and these were linked by some kind of disulfide bond, or you can say a covalent bond. And that's why on SDS page, in presence of beta methanol, you are getting a single band, that of 25 kilodalton. Going to the next question. Here the concentration is given, and you have to identify the free energy change under the given physiological conditions. The formula which would be applied would be delta G is equal to delta G0 plus 2.303 RT log of the products, because what you have to do is, here what you have to calculate is free energy change for synthesis of ATP. So reaction would be ADP plus inorganic phosphate is going to give you a ATP. So RT products are ATP, ADP, and products is ATP. So product is ATP divided by, it would be ADP by inorganic phosphates. Now simply you have to put a value. The value of delta G0 is given, but this is the delta G0 of ATP hydrolysis. So it is the value of ATP hydrolysis. So ADP synthesis, this would be, value will remain same, but it will be plus. So here you will put a plus sign, 30.5, plus you put over here values of this 23. R value is given, that is 8.315. T is given, that is temperature over here is given somewhere, it would be given temperature 37 degrees centigrade. So in Kelvin it would be 310 Kelvin. Log of ATP concentration is given. Log of ATP concentration is 2.59 millimolar, that is 10 raised to minus 3, divided by the values of 0.73 into 10 raised to minus 3 multiplied by another is 2.72 into 10 raised to minus 3. If you solve this equation now, finally you will get an answer of 18521.2 kilojoule per mole. So that will be the correct answer. So you have to simply put this equation for solution for solving this numerical. Question number 54, this is straightforward question. An algorithm was designed to find globin in a protein sequence. So there is some kind of protein sequence database. There are 78 globin sequences, and out of 78 globin sequences, 72 sequences were retrieved, where 65 were globin. So in other words, from 78 actually 65 globin were retrieved. So you can find the sensitivity. Sensitivity will be 65 out of 78 into percentage. The value will come out to be 83.3. So the total was 78, and out of this only 65 sequences were being fetched and were similar to the globe. 65 for the globin. If the sensitivity would be we can say 100%, then all the sequences should be of a globin. Then question number 55, this is also a very simple question, where what you have to do is, you have to calculate the free energy change associated with the transfer of electrons from NADH to oxygen. So the formula for free energy change, delta G0 would be equivalent to minus NF delta E0, and for E0 would be E of final minus E of initial. And during this reaction, what we have to do is, when NADH it delivers its electron to oxygen, you have to calculate. So E final would be the oxygen. So value of this is 0.816 minus initial is of this, that is minus 0.32. If you solve this, what you'll get is 0.816 plus 0.32. The value what you will get over here is 1.136. And now you can apply this equation. There are 2 electrons which are flowing, because NADH it delivers 2 electrons which passes to the oxygen. The value of Faraday constant is also given, I think it would be given somewhere here, and even if it is not given, we can remember it, but it is given. Okay, it is 96,500, and potential of this is also given, and potential we have calculated, it is 1.136. If you multiply it, here you will get the value of minus approximately 219,248. That would be joule per mole. If you have to convert to kilojoule per mole, it would be 219,248 kilojoule per mole. That would be the answer. So in this way you can solve this particular numerical. So these were the questions of biotechnology GATE 2020. Again, subscribe, do subscribe our channel, so you can get an alert. We are going to upload various free examination solutions, as well as we are also going to put various demo lectures on this YouTube channel. You can use them for your preparation for different examinations. Once again, thank you. Like our channel. You can visit our website ifasonline.com. Again, thank you and signing out. Bye bye.